This video is brought to you by Honeycomb Technologies. The pufferfish, also known as blowfish, is a tropical fish found in the warm waters of South America, Central Africa, and Southeast Asia. It is characterized by its round appearance and known for its defense mechanism, filling up its bladder with water to avoid predation. Animals that are unfortunate enough to bite on a pufferfish end up overwhelmed with an unpalatable pointy ball. The pufferfish also has another line of defense. The body of a pufferfish is inhabited by bacteria that produce a deadly, paralyzing toxin called tetrodotoxin. Animals that ingest a blowfish will find their stomachs full with tetrodotoxin, which can kill many animals in less than 1 hour. Tetrodotoxin is one of the most potent neurotoxins in the world. It is about 12,000 times more poisonous than cyanide. Pufferfish meat is considered a delicacy in Japan. Here we can see pufferfish being sorted in a Japanese fish market. 10,000 tons of blowfish are consumed each year in Japan. The preparation of the Japanese fugu meal must be specially prepared by certified chefs to avoid human consumption of the deadly neurotoxin. This is life or death for people eating a pufferfish meal. A single wrong cut means death for a customer, as a single puffer has enough poison to kill 30 humans. Restaurant preparation is restricted by law in Japan and several other countries because of the dangers that come with handling the fish. The meat of the fish is sold openly in markets. However, whole fish are not available to the average consumer. The meat must be prepared for the customer on site by a licensed fishmonger. In order to obtain the license to prepare fugu, applicants must complete a 3-year apprenticeship, followed by multiple written exams and finally a preparation test. Only about 35% of applicants pass. Despite the rigorous standards that fugu chefs are held to, the Tokyo Bureau of Social Welfare indicates that an average of 44 fugu poisonings occurred per year between 1996 and 2006. The potent neurotoxin tetrodotoxin shuts down electrical signaling throughout the body by blocking sodium ion channels in nerve cell membranes. The result is a total paralysis of the muscles. However, the victim remains fully aware. Left untreated, the victim will eventually die from asphyxiation. The severity of poisoning cases varies significantly based on the consumed dose. However, 100% of cases require hospitalization. Death from tetrodotoxin poisoning from pufferfish consumption is low, with only a 6.8 fatality rate. However, poisoning cases cause lots of negative publicity for high-end restaurants and cost adventurous eaters a large unexpected hospital bill. Despite the risks of eating fugu, the dish has remained popular in Japan for centuries. About 40 kinds of fugu are caught in Japan, and the people consume 10,000 tons of the fish every year. In recent years, it has made its way into upscale American dining. As of 2003, only 17 restaurants in the United States serve fugu. Of the 17, 12 were located in New York. Strict limitations imposed by the FDA limit the import of fugu, mainly that all the tetrodotoxin must be removed from the fish prior to its entry into the United States. All this required pre-processing of this fish only increases the price and decreases the freshness. Here at Honeycomb Technologies, we have developed a groundbreaking technique to guarantee that customers are always provided with a 100% toxin-free fish through triplex DNA silencing. The metabolic synthesis pathway for tetrodotoxin is still a scientific mystery. Pufferfish toxicity shows remarkable individual and regional variations. TTX is distributed over various organisms, including food animals of pufferfish. The trumpet shell accumulates TTX by ingesting toxic starfish, and marine bacteria primarily produce TTX, suggesting that pufferfish do not synthesize TTX but accumulate it through the food chain, which starts from marine bacteria. If this is the case, pufferfish should become non-toxic when fed TTX-free diets in an environment in which the invasion of TTX-bearing organisms has been eliminated. Initial studies based on this concept have been performed successfully, but on relatively small scales. Fish produced when fed non-TTX containing diets were significantly less toxic. However, only 40 fish out of the 4,133 specimens from the net cage culture and 910 specimens from land culture proved completely devoid of tetrodotoxin. By using triplex DNA silencing to eliminate the pufferfish's natural immunity to its own poison, we could guarantee that every fish that we provided to our customers was 100% safe by eliminating toxic fish from the population without any post-processing. This would not only allow customers more peace of mind when consuming this culturally significant fish, it would also open doors for many more restaurants to consider serving this dish. Restaurants in the United States and internationally would be interested in our product because the expensive licensing process would no longer be necessary. They could now sell this expensive delicacy without the risk of poisoning customers, and there would be much less bureaucratic intervention when it came to imports and exports. Sodium channels are essential to the proper function of nerves in conducting our movements. Pufferfish's innate immunity to TTX comes from a special mutation in domains 1 and 2 of their sodium channels that at a molecular level significantly decreases the affinity of TTX binding to the pufferfish's own sodium ion channels. From the amino acid sequence of Fugu sodium ion channel, we can determine the actual gene sequence, shown here. By silencing the codon sequence AAT that corresponds to the TTX-resistant asparagine addition, we can eliminate the conformational protein blockage that prevents TTX binding in the nerve cells of pufferfish. We use the factors bind the silencing enzyme stably to ATT site in the genome. The DNA randomly moves around and forms transient loops. The enzyme hits other regions in the genome nearby, which then become modified and are switched off. Now, since the pufferfish's natural immunity is gone, if there is any buildup of the TTX in the pufferfish, it will block the puffer's sodium channels using the same mechanism through which it poisons predators.